Thank you to the organizers for the opportunity to present my work today. My name is Connor, and I am a Washington Research Foundation postdoctoral fellow in David Ginger's group at the University of Washington. Today I'm going to talk about what happens when ions are electrochemically driven into conjugated polymers to form what we call these polaron-ion pairs, and how these polaron-ion pairs move through the polymer and how the polymer structure changes when ions are injected or electrochemically driven into these conjugated polymer materials. And so one of the main motivations for this work is that many different technologies rely on what we call organic mixed conductors. And so these are materials that can transport both electrons and ions. And they've been used a lot in bioelectronics applications. So for instance, as organic electrochemical transistors, which I'll talk about a bit more. Also, in all polymer charge storage with an aqueous electrolyte, they have the advantage of being very fast to charge and discharge, and in other applications including neuromorphic computing and thermal electrics. So the organic electrochemical transistor is a device that we focus a lot on in the group. So these devices have a lot of advantages over kind of the state-of-the-art bioelectronic devices that are typically made of silicon. So first of all, the active layer is solution processable. You can dope these active layers volumetrically. You have chemical versatility, and they're also biocompatible. And so if we could zoom in on this active layer in this transistor here, we would see what's happening is that when you apply a gate bias, ions are driven into this polycrystalline conjugated polymer layer to form these ion hole or ion electron pairs, which then changes the conductivity within this channel. And if we then apply a reverse bias, the ions come out and the conductivity goes down. And so that's how this device switches on and off. And so these devices have been used a lot for different bioelectronics applications. This one example recently from our group where we stuck one of these devices on a Venus flytrap. And so here you can see that as we mechanically stimulate the inside of this flytrap, you can record signals with this organic electrochemical transistor quite easily. And so the big challenge with these devices is really a fundamental challenge. And so not a lot is known about how ions are injected into these types of materials and the properties of these materials that change once ions are injected. And so this is just a quote here from George Malieris in a recent Nature Reviews Materials paper. It says, little is known about ion injection from electrolytes into organic semiconductors owing to the affinity in measuring ion injection in the presence of an electron charge. And what he's saying here is we can make these devices, but we don't really understand the fundamentals of what's going on during their operation. And so this is something that's really important to understand so we can develop new design rules for making new devices with enhanced properties. So a major challenge in fabricating these devices is that polymers aren't typically designed to transport both ions and electrons. And so these are just two examples here of P3HT and PBTTT, which are polymers commonly used for things like organic field effect transistors and photovoltaics. And because they have these alkyl side chains, they're really bad at transporting biologically relevant ions. And so what people have typically done about this is change these alkyl side chains to polyethylene glycol side chains. And what this does in a paper by Jonathan Rivnay and Alex Giovannotti is changes the doping mechanism. So instead of just having a surface level of doping, you actually get ion injection into the bulk of the polymer. Also recently, we've shown that if we glycolate the side chains of a polymer like P3HT, we get much faster ion injection into the polymer. these materials that I'm going to be talking about today are conjugated polymers that are used as mixed ionic electronic conductors. And so they have typically side chains that enable ion transport, like these ethylene glycol side chains shown here, and also a conjugated backbone that enables electron or hole transport within the material. So we start by looking at two polymers that have very similar structures. The first is PB2T-TEG, and the second is P3MEEMT or P3MEEMT. Both of these polymers have conjugated backbones and these ethylene glycol side chains, but they actually behave quite differently when ions are injected into them. And so the kind of questions that we want to ask about this process of ion insertion is, how does this polaron-ion pair move within the material? And also, how does the formation of this polaron-ion pair impact the polymer structure? And so the kind of spoiler here is that we find out that PB2TTEG undergoes a first order structural phase transition upon ion insertion, and P3MEEMT has a very minimal structural changes upon ion insertion. And so to answer this first question, how does the polaron-ion pair move within the material, we perform a fairly simple experiment. And so what we do is we take our sample, this is a polymer thin film just on ITO, and we dope half of the sample. So we have ion polaron pairs on one side, and we leave the other side undoped. And then what we do is we use an optical microscope to watch this interface relax back to equilibrium. And so what we would expect if these polaron ion pairs can freely diffuse is that the diffusion can just be based on Fick's second law. And so in this graphic here, what we would expect to see in the sample is a blurring out of this interface as these polaron ion pairs move into this undoped region. And so here's the result of this experiment. So first I'm going to show the result of P3MEEMT. And so if I play this movie here, you can watch how this interface moves with time. And you can see that it behaves basically as we expected it to. So aside from some immobile fraction of these ion polaron pairs, we see that this interface does blur out over time. If we look at PB2TTEG, on the other hand, what we see was really surprising to us. So instead of this blurring out of the interface, what we saw is that the interface remains sharp as it propagates. And so here you can kind of compare, these are just line cuts of the two results, and you can see that they behave very differently. And we can perform calculations to model these two diffusions. So this is what you would expect for Fickian diffusion, which looks a lot like P3MEEMT. And if we restrict our diffusion just by having two different diffusion constants in the undoped and doped regions, we get something that looks a bit more like PB2T-TEG. And so we wanted to see exactly how sharp this interface is. So instead of using optical microscopy, we switched to an AFM-based technique called photo-induced force microscopy. And so this is a technique that is really just similar to an FTIR spectrometer, only it has about 10 to 20 nanometer spatial resolution. So I'm not going to get into exactly how this works, but by using an infrared active ion, such as PF6 here, we can map this ion transport with really high spatial resolution. And so this is just the PIFM spectrum we get here. And you can see that, and we monitor this PF6 peak here around 850 nanometers. And so if I play this movie here, we can see how the ion concentration changes with time as this front moves across. And even at really high spatial resolution, this is a very sharp interface between the doped and undoped regions. And you can see it kind of in this field of view, you don't see much change in the topography. So if we zoom a bit more, so now we're looking at a field of view that's only about 3 by 3 microns, and I played this movie again. Now you can start to see that this ion migration through the film is fairly heterogeneous. And so we're still exploring this a bit more to try to understand how ions move through crystalline and amorphous regions of the polymer. But what was really cool about this result is that we could see changes in the topography as this ion front moves across. And so the presence of the ion is actually changing the topographical features of the polymer. And so it's a bit hard to see in this movie here, but if I go to the next slide, here I'm showing, instead of the topography, the change in topography with time. And so if this movie plays, now you can see that there are major changes, both positive and negative, in the topography as this ion front moves through the film. And so this was really exciting to see because we could get a nanoscale picture of how ions move through soft materials like this PB2TEG polymer. So to sum things up so far, we have these two different polymers that have very similar structures with these glycosylated side chains and conjugated backbones, and the polar and ion pairs in these materials diffuse in very different ways when we make this interface. And so now we want to try to understand what is the origin of this diffusion in PB2TEG, and why does it look so different than what we anticipated? So before I get to PB2TEG, I want to show the typical example of how a conjugated polymer crystal lattice changes upon ion insertion using X-ray diffraction. And so the polymer most commonly studied is P3HT, which is shown here. This is really the fruit fly of conjugated polymers. And so in this paper by Chabanik and Siegelman, what they show is that as you insert ions into P3HT, or as you drive more ions in by increasing the voltage, you see a continuous expansion of the P3HT crystal lattice. So the more ions that go into the polymer, the more this crystal lattice expands. And so this is really the typical picture of ion insertion into conjugated polymers. The more ions, the more the polymer expands, and this expansion is continuous. And so this has also been seen by other groups. So inorganic materials behave in a much different way than these conjugated polymers. And so here's an example with iron phosphate, so a very typical lithium ion battery material. And so here, as we lithiate iron phosphate, you can see that we don't see peak shifts. What we see are new peaks appearing. And this is indicative of a structural phase transition as the lithium enters this material. And so a recent study has also shown that this results in very sharp phase boundaries between lithium-rich and lithium-poor regions of these materials. And so this made us think that maybe something similar is happening with PB2TEG. And so the different mechanisms for ion insertion into these materials has really big implications on how these materials behave as charge storage devices. And so for lithium iron phosphate and iron phosphate, these materials have a very flat charge and discharge curve, meaning that the voltage stays constant as the material lithiates and delithiates. Conjugated polymers, on the other hand, like this polypyrrole, have these triangular shapes that are indicative of supercapacitors or pseudocapacitive behavior. And so I'm just going to set this aside for now, but I'm going to come back to this later when talking about PB2TEG. All right, so now back to these two polymers. So we did these experiments, these X-ray diffraction experiments on both P3MEEMT and PB2TEG. For P3MEEMT, as we increase the voltage and drive more ions into the polymer, we see that the diffraction peak doesn't change much. It expands maybe a little bit as we add more and more ions. For PB2TEG, we saw something that was actually really surprising to us. So we see initially we start off with an expanded or a contracted crystal lattice, and as we add more and more ions, suddenly it expands over a very small voltage range. And so you can see that here we start off with a fairly small lamellar spacing, and between 0.2 and 0.3 volts we see this huge jump and the lattice spacing. And so it's also important to note that this effect is reversible. So we observe this reversible kind of discontinuous lattice expansion in this material. And so to get more or higher resolution patterns of this material, we performed grazing incidence wide angle X-ray scattering. And we looked at both 0 volts, so before this discontinuity happens, and around 0.4 volts. And what we saw that was really cool is we see two very different diffraction patterns at these two different voltages. And so these look almost like two completely different polymers, but they're actually two distinct crystal structures of the same polymer. A question that was really interesting to us is really what's going on in this discontinuity. Do we see kind of a smooth shifting between these two crystal structures like we saw in P3HT, or do we see the presence of both of these crystal structures like we did in lithium iron phosphate? And so what we actually see is a linear combination of these two different crystal structures, making us think that we actually have phase separation between ion-rich and ion-poor regions, and that this polymer is going through a first-order structural phase transition. So the previous GWAX experiments that I showed were all actually on dry films that we basically dope an aqueous electrolyte, take out of the electrolyte, and then measure. But we wanted to see if this effect was also happening in situ. And so what we did is we constructed a custom sample cell to do GWAX in an aqueous electrolyte. This was more challenging than we thought because you need really, really small angles in order to measure these diffraction peaks. And so using this liquid cell, we were able to see that this effect also happens in situ. So you can see here as we increase the voltage, we see one peak disappear and another peak reappear at a much expanded lattice spacing. And so comparing our in situ to ex situ results, we see that they look about the same, only you see this kind of shift in the spacing. And we think this is due to some swelling in water. And so the kind of summary here is that this effect also happens in situ in an aqueous electrolyte. And so coming back to these dry results I showed, we worked with Ying Dao and Daniel Davies at UIUC to solve these crystal structures. And we hypothesized that the mechanism for what's going on in this polymer is alternating between side chain crystallization when it's undoped and backbone crystallization when it's doped. And so one kind of simple feature of this is that we don't see a pi-pi stacking peak in the undoped state, and we do see a polymer backbone pi-pi stacking peak in the doped state. And so we think that this polymer actually acts like a molecular zipper. And so ions can come in and basically unzip this polymer, causing the side chains to not be crystalline anymore and crystallizing the polymer backbone. And so it was really exciting to see from these diffraction patterns that we could hypothesize this mechanism. And so the next question is really, what is actually going on on the nanoscale with this polymer? We know we have some initial ion insertion. We know in the middle we have phase separation, and then we have this final expanded crystal lattice. But what does this phase separation look like? And so to probe this directly, we again used our microscope, our AFM photo-induced force microscope. And this is the kind of data that we get. And so this is just adding more and more ions at each frame and watching how this polymer expands. And so based on this movie here, you can see that as we add more ions and inject more ions into this polymer, certain areas are expanding, whereas certain areas are not. And we can correlate this directly with our photo-induced force microscopy signal to pinpoint exactly where these ions are, and we see that they correlate fairly well with areas that are expanding in the polymer. And so if we, instead of looking at the topography, we look at change in topography, now you can really see that there's a really good correlation between the local ion concentration and the change in the topography. And so this has allowed us to really visualize what this phase separation looks like at the nanoscale. And we're still trying to understand exactly, why certain areas are expanding and certain areas aren't. Is it just a kind of nucleation and growth type stochastic model, or are there areas of the polymer film that are actually preferential for ions to be inserted and for this expansion to happen? So this is still kind of an undergoing investigation of what's happening inside this polymer film. Okay, so so to kind of sum things up, now we know that we have our P3M-EMT. It has this diffusion that looks Fickian because there's no lattice expansion. Yet PB2TEG, it has this sharp interface, which we believe is due to the fact that the phase transition is actually limiting ion diffusion in the film. And so in the top case, we have P3M-EMT, where ion diffusion is just limited by the intrinsic diffusivity of the ions, where in PB2TEG, we see that the phase transition is probably limiting ion diffusion. And so the last question that we wanted to ask is, what does this charge-discharge profile look like in these conjugated polymers that have this phase transition? Is it flat, like we see from inorganic materials that undergo phase transitions, or does it look more like a supercapacitor? And we weren't exactly sure what to expect, but we actually get something that looks like it's kind of in between these two cases. So this isn't a perfect triangle, but we also don't see a perfectly flat plateau. And so this is still an open question, why do we see this behavior? But it looks like we have kind of one slope when we have this phase separation, and then once this phase transition goes to completion, we see more behavior that's like a supercapacitor. All right, so to sum things up, we think we've discovered a really interesting phenomenon that happens in this polymer PB2TEG, where it has completely different crystal structures in the neutral and oxidized phase. We watched this polymer, ions move through this polymer with nanoscale resolution and watch the kind of initial doping processes in this polymer. And last showed that it has this very unique charge-discharge behavior. And so I think that this phase transition has really big implications for how devices using these materials will operate. And so the future is really, can we exploit these transitions in organic materials to enhance the performance of things like organic transistors or organic energy storage? So last, I would like to thank David for being a great mentor. Everyone bolded in this list works on ion transport in our group, and then our collaborators, Christine Luscombe's group, who made the P3M-EMT, Chengxing Li's group, who made the PB2TTG polymer, and Ying Dao's group, who helped us interpret some of the GWAX results. And my postdoc is funded by the Washington Research Foundation, and other funding is through NIH, DOE, and the Mistletoe Foundation. And I'll be happy to take any questions.